and some of them are also faculty members from different universities in the Philippines. And of course, we have participants from Taiwan in the person of Dr. Machida. And I guess there are more from other countries. Sometimes we get participants from Dubai. So it's quite interesting. Our reach now is, we are expanding a bit.
It's great that you are extending all that area, because it's a very
Yes.
area that is in need of these things.
Mm-hmm.
Yeah, I mean, it's more convenient because like we can do online.
Hello.
Ryuji says hello.
Hello, Ruyi. Good to read you, man.
Yeah, I think we can start and then like just let people come in as we proceed.
Yes, we may start now.
Can you see my screen?
Yes.
Okay. Hello, everyone. So I'd like to welcome you to the last lecture for our series for this year. So we're now in our third year of Philippine Planktology lecture series. And this is in collaboration with the University of the Philippines, Visayas, in particular, the Institute of Marine Fisheries and Oceanography, together with the Plankton Society of the Philippines, the Philippine Planktology, and the Plankton Niche Lab at the University of Visayas. So our speaker for today is Dr. Leocadio Blanco-Bercial, who's currently affiliated with the Bermuda Institute of Ocean Sciences and the Arizona State University. So for the entire program of our lecture series, I'll just have a quick recap of the house rule, and then I'll go immediately with the introduction of Leo. We'll have the 30-minute allotted time for the proper talk, and then there will be allotted time for the open forum wherein the participants are allowed to raise their questions. And then we'll proceed with the awarding of certificates to Leo and then some photo opportunity if we still have time. So for the house rule, since we're doing this online, participants are requested to turn off their cameras and keep their microphones on mute so that we will be avoiding this disrupting the entire lecture. Please hold your questions up until the end of the presentation. Questions will be entertained during the open forum after the talk. To ask for questions, you can raise your hand so that you can call our attention and then let you talk. And then you can wait for the moderator to call you in. You may also type your questions in the Q&A box. Please do not spam the chat box as we want to get more all of the questions as much as possible. So kindly accomplish the evaluation form at the end of this talk so that you can claim your e-certificate after. And for the quick introduction of our speaker for today, so we have Dr. Leocadio Blanco-Bercial. As I've mentioned, is currently a researcher and assistant professor at the Bermuda Institute of Ocean Sciences and the Arizona State University. Leo received his PhD from the University of Oviedo in Spain, where he studied plankton ecology and population dynamics. Afterwards, he did his postdoctoral fellowship at the University of Connecticut, I think under the supervision of Dr. Anne Bucklin.
Mm-hmm.
He added molecular tools to invest within his expertise to investigate the spatial and temporal patterns of distribution, connectivity, and evolution of marine plankton. He joined Bermuda Institute of Ocean Sciences in 2015, and with the integration of BIOS, or the Bermuda Institute of Ocean Sciences, and the Arizona State University, Leo is now an assistant professor at the School of Ocean Futures and the School of Life Sciences. His research is mainly focused on understanding how eukaryotic diversity is originated maintained, and organized in open ocean. So in most of his research, he usually integrates molecular and classical tools to address questions related to plankton community diversity and impact on present conditions and future changes in the ocean. So without further ado, I'd like to give the floor to Leo for his presentation.
All right. Thank you very much. And thank you very much for inviting me to come to give my talk. Okay, share screen. Share. Perfect. I don't know if you would like to address, there is a raised hand. I don't know if you would like to address it before we start. Louis?
Yeah, we can see your slides and it's all working.
No, well, there is a raised
Oh,
hand
raise hand? Okay.
before we start. You might want to, so maybe you would like to address the raised hand before I start.
Yeah, I think you can start anytime. Yeah,
Okay,
you can go
okay,
ahead.
don't worry then. All right, well, first, thank you very much for inviting me. I'm going to talk about the plankton diversity in the high throughput era. And I mean about the high throughput era of data, huge amount of data coming to us. Then how can we analyze it better? First, I would like to thank my research assistants, Hannah, Kevin. Kevin is not in our lab anymore, but well, he originated part of the data. And yes, they are the people that are actually in the lab doing a lot of the handwork of the samples, processing the samples. Then they send me the data and then I do the analysis, etc. But we have to always acknowledge the people that are in the labs creating all the data. We are a team, and this is like my larger team that includes, I'm going to put here a pointer, a laser pointer. Well, myself, that is one of the PIs, Amy Mas, that is actually another PI in the, let's say, two labs that they work very close together. Also, we are married, so we are really very close to each other. And then we have Hannah now and Jess, or two research assistants in the lab, together with the three graduate students that we hired, Lauren, Rocio, and Sem Dosegal. We're a very international lab from Spain, Bermuda, Latin America, US, the Netherlands. So actually it's a very diverse, interesting accents everywhere in the lab. So anyhow, we are a team. A lot of the data you'll see is a product of a large team of people. Also, I'm the one presenting it. So first, what is biodiversity and what I'm going to be talking today? Isn't it biodiversity? This is one of the general definitions, variety of different types of life found on Earth. It's a measure of the organisms present in different ecosystems. So this is the United Nations definition. This is a very common one accepted. But then one of the things we have to think about is why this is important. Why is biodiversity? Why should we care? Well, biodiversity provides ecosystem services. So we know that it comes tourism to see your coral reefs, your mountains, your forests, fisheries, more diverse ecosystem usually host more species and more biomass. Pharmacology, all these crazy natural products taken from sponges or whatever in the ocean. Actually, these are things I'm sure you are very aware of the Philippines, since you are in one of the biodiversity hotspots, both inland and in the ocean. The theory is that highly biodiverse ecosystems are more stable facing challenging conditions. They provide resilience against changes because there are more species, they might have more traits, different traits that will allow them to, well, one of them or more of them will be able to face these changing conditions. But also is beauty. So there is a study and they prove that as humans, if we take the same biome, we think that, or we perceive biodiverse biomes as more beautiful. So I would say Bermuda has great reefs, for coral reefs, very well preserved, but they are low diverse. They have very low diverse because we're a cold reef factory. We're very north, we're like 30 degrees north. Most people that come from places like Philippines, Indonesia, or the Caribbean or Red Sea, they come to Bermuda and say, well, this is like a boring, not very beautiful, despite that maybe the conditions, the health of the coral reef is higher in Bermuda just because we have a lower biodiversity. Okay, so but then these are questions of why biodiversity is important. There are many more, of course. But since I work with plankton, I'm going to talk about the plankton diversity in the high throughput era that I was talking about. This is basically just zooplankton. Today I'm going to be focused just in metazoans, but this is just pictures that I have taken during my years, most of them here in the Sargasso. Here we have annelida, arthropods, many different groups, mollusks, chordates. We have chitognats, Cnidaria, both here, for example, this beautiful siphonophore, but also here growing on top of the theropod. So we have many different phyla living in the zooplankton all at once, making one of the clearest examples of high biodiversity environments. And I'm going to talk about in the Sargasso Sea, that is also one of the biodiversity hotspots in the world. Then, when it's great to be in a place with such a high biodiversity, but then how can we measure? How do we measure it? Because there are a lot of species and a lot of individuals when you take the net. This is a hyperdiverse group, so then you look at the animals and they look all different. They have a highly complicated taxonomy with cryptic species, with things that they have not been described, etc. So how can we do it? And before I go into this talk, when I arrived to Bermuda, I look at all the literature that existed about the region. And there is this paper by Georgiana Divy and Brooks in 1977. She's the one that likely did the most detailed work in the taxonomy of the zooplankton in Bermuda. And then I was reading the paper, and in the introduction had this excerpt that is, I'm going to read it because when I read it, I was like, oh my God, this is going to be very difficult. Because of the taxonomic problems and amount of time that will have been involved, no attempts have been made to identify many small species of such genera, such as, for example, Paracalanus, Clausocalanus, Spinocalanus, Microcalanus, Escolithitricella, Scaphocalanus, Ethionankea. Also, some larger species were not identified. So likely, she couldn't identify two species, any of the small copepods. So these few genera that she's mentioning likely includes over 100 species, and likely she means many other more genera of the same groups. What we call the Clausocalanoidea is a hyperdiverse superfamily in the calanoid copepods. Even some larger species she couldn't identify. So likely she left behind hundreds of species without being identified. And still, she was able to put them to more than 300 species just for calanoid copepods. Forget about many other things like Cyclopoida or any other things that she couldn't. So that means that likely here in the Sargasso, there are over 1,000 species or way more than 1,000 species, but likely we don't even know which ones they are. This is a picture that I took from just one day. I took a scoop from sample, not prepared, and just took a picture with the camera of the microscope. And then when I was looking at it later, I realized that, well, there are some big animals. This is Pleroma massiphias. This is one of the workhorse, one of the animals that we use for our experiments, some big annelid, amphipod that we all recognize. But then of all the other copepods and ostracoda you see here, well, this is, for example, Cnidarian larvae. Of all the others, likely maybe one or two of these, they are repeated more than once in the picture, or a few. So that is giving you the idea of how diverse this community is. Then how can we approach it, since the microscope is going to be almost impossible or will take too long, isn't it? I will not get funded by any agency to spend 6 months in a microscope. So we are using this technique called metabarcoding to get an idea of the taxonomic and phylogenetic diversity. What we do is we use a short region of DNA from some specific gene to identify all the species present in a sample at once. What we do is we amplify that small region, and we do this high throughput sequencing. Basically, we throw all the DNA that is originated from that sequence into a machine that is able to put them separated in a small plate of glass, let's say, and then an image, a camera is able to identify each of them when they are being amplified. Then we can take apart all these different DNA sequences and then count how many of each of these different sequences there were in the sample, and it forms the identity and the phylogeny, isn't it? This is a technique that exists already I'm afraid I'm getting old, for almost 20 years. And now it's pretty much established. I suppose actually it was covered by some of the previous topics in this in this series. But again, we study the DNA, we get the genomic DNA, and from the amplification, the PCR, the DNA, we get a proxy of taxonomy and relative abundances. But with this method, we don't get biomass or any of the shape, let's say. That what we cannot, and if we don't have a reference for the sequences, we don't know what is there. So we do also a second set of analysis with the same samples. That is, we put them in something we call the SoScan. It's a high quality scanner that is in a box that make it waterproof, so the scanner doesn't get ruined when we throw inside the sample. Then HANA is this hand, separates every single individual from each other. We put around 1,500 individuals here, 1,500. And once they are all separated and touch with each other, we scan it. It's a high quality, high density, 4,800 pixels per inch. And then we have a huge several gigabytes image that then we process it. This is just really software doing it. And it's going to separate each of the shapes into a separate image. Then through actually in artificial intelligence, machine learning, we pass it through a training set that is able to say, okay, this looks like krill, this looks like a copepod, this looks like whatever. And it's able to classify all of them. Actually, it's good only like 70% or 75, 80 if you are not in a very complex area. But then you have to spend time validating what the machine has done. That's a lot of time consuming. But then we get this imaging analysis because this instrument is going to measure length, width, how dark the animal is. For example, this is important gray level. I'm going to be talking about later, how transparent, etc. How does it have a lot of appendages? Is more or less round? So all these things. And this is what we are using now to use some kind of a numerical approach to diversity. So then, since again, it's extremely tedious with microscope, so then we have these semi-automatic methods. So the first question I wanted to ask is there a seasonal cycle in the Sargasso sea in the zooplankton community? And is it linked to some basic biochemical cycles, such as carbon export that we're all measuring? What is happening with the carbon in the oceans? All right, so actually such a simple question has not been really answered any time before because, well, the community is extremely difficult. Here there is a very basic graph between, this is the carbon export, the flux, okay? And it here in this dotted line is the number of species that we have measured by metabarcoding. And we see that there is no clear relationship. There is always this between 700 OTUs or taxa or whatever you call it, ASV, single variants. Let's call it a species or proxy to species. Through the year, between 700 and 750, but then the carbon export is coming up and down, up and down. So doesn't look like there is any relationship. So what happens if we do a very basic analysis of taking these proxy to species that we have by the relative abundance, and we calculate the beta diversity, the similarity between the samples, and we organize it in a two-dimensional graph that basically does is they put them closer to each other, the samples closer to each other are more similar. So in this graph, that is a non-metric multidimensional scaling, MDS based in a similarity. This is very Curtis, but almost with any other similarity, statistics will work. What we find here is that the closer two samples are is because the communities are more related. And we have a beautiful seasonal cycle starting by February, March, April, May, June, July, August, September. In October, there was a hurricane. So forget we couldn't sample November and December. So we can see a very nice arrangement of the samples in the seasonal cycle. And actually the colors that you see here, they are actually different statistical significant clusters of species. So what we have here is actually the winter mixing, when we have this the spring, when they start to have you have the bloom, then we have the transition into the highly stratified summer, and finally the winter mix, the fall mixing into the winter. Okay, so we have that. And what we found to doing some basic analysis, there was a significant correlation with the sported flux of carbon, nitrogen, and phosphorus. So perfect. We have the seasonality, and we see that actually the community of zooplankton is related to the carbon export, that actually there was not a good relationship before. I look at the images that we have in the scanner, and it looked like, yes, when we have a lot of flax that is in the spring, if you remember, so these are the representative images for each of these 4 seasons. And when I plot it against the carbon flax, I was like, okay, this is great. When there is a lot of flax, we have vertical migrators, such as the krill or the big peromamas, etc. And actually some animals that create big houses, Eucopleura, they create some houses of cellulose that they live inside, and then they discard this house when it becomes dirty. And that we know that all these animals, they causes a lot of flax. So, all right, with the images, we can see that this looks like is what is happening. Then when we start to go through the summer, we move into different groups. Kalani, the other group of copepods that we know that they don't have such a strong diurnal migration. And the community, actually, what that happens is that becomes dominated by the species that are more and more indicative of recycling. They are not just feeding in the primary producers and taking it down. They're actually feeding on each other, very complex food webs in surface, that what causes is that the export is not as strong. So, all right, I was very happy, but this is not very numerical, isn't it? So now what we did, that is a paper that is actually in review, have just minor reviews, hopefully will be soon out. This is a master by a PhD student now in Sorbonne, Margot Perraga. I was part of her master's committee and part of her PhD committee. And what we did is, okay, let's take a yearly cycle of both the scans, for the source scan and the metabarcoding, and let's try to integrate both kinds of data. Instead of just comparing, let's say, can we do some numerical approach to put both together? Because this is what we are all trying, it's not just us. So the first thing that we do, the same thing as you have seen before, let's take the molecular data and see, do we have any kind of change in the community composition? So this is similar to the MDS that we saw before. This is a principal coordinate analysis. It's a different kind of 2D ordination of the samples, in which the samples that are closer to each other, or they are more similar, that are closer to each other. Also, C built just a tree that represents how similar the species are to each other, and statistically she organized them into actually 3 different groups. That will be the spring bloom, what will be the winter or the fall and winter, and will be the summer stratification. Bermuda, similar to the very hot places, in summer you have this thick wood layer on surface in summer, like from surface to 50 or to even to 80 meters. Very warm, very separated from surface, and that changes everything. So, okay, I have this kind of way to compare or to organize my samples in the molecular data, but this is something similar to what I have already done. So what happens if we use these groups and we apply them to organize my image data from the metabarcoding? And one very simple approach, I'm going to change my picture here. Sorry, I had it in the right end. I was not seeing the figure properly. I was talking to you before about transparency of animals. All right. So here in the bottom axis, in the bottom figure, what I have is the major axis, how long the animals are. We know that size of animals control many of their biology because of this allometry. Solarian animals breathe slower, etc. Isn't it? But what we can see is that actually there is no much change in the shape of that curve, isn't it? This is the distribution of the sizes in each sample. The densities is you have the size, larger animals here, and this is how many you have. And actually, the 3 curves, they are not that different, isn't it? They have the mode more or less in the same place. It's true that there are more animals in spring, in bloom, because we have more food, but they are not terribly different. But where we found the big difference actually was in the gray level. The gray level is one value, this is the standard computer way or image way to see color, in which 0 is completely black and 255 is completely white. All right. So what we found here is actually during the spring blooms, animals are actually darker, all right? There are lower values of gray level means darker. When during the summer and the spring, we have more transparent individuals. And that is very important because it's indicating that there is a change in the community that actually we cannot know only if we look at these sequences, isn't it? This is a very dramatic change into having darker individuals during the spring bloom. And so we followed that. And what we found, actually, if we compare for all the samples that we have, all the lengths, and we try to compare it to what happens with the carbon exports in the y-axis, for example. So here is the axis or the perimeter. So all these are measurements related to the shape and the size of the individual. Compared then to the carbon export, none of this is significant. So actually the size of the individuals is not affecting the ability of the community to export carbon. However, when we look at the different levels of the different measurements of transparency, actually we did find that this community actually, the darker it is, the lower the gray levels, the more carbon export we have. And this is great because then now we know what we have to look at. What are these individuals that are transparent? They are decreasing the export, or which are these ones that are very dark? They are increasing the carbon export. Essentially, they are feeding in surface and then taking whatever they are feeding into the deep and removing carbon into the deep ocean. Other things that we are trying to do is, for example, this is what we call ASV modules. What we do here is we take all these single proxy to species that we have in the metabarcoding, and we look which ones occur at the same time, which ones co-occur over time, which ones are associated with each other. And when some of them, they are always appearing at the same time, we call it a module, and they go together. So here, for example, you have a gray module in the back, this green one in the front, or the black, this dark gray. So these are different modules, different groups of taxa that they co-occur. But also we have from the images, we can distribute the images based how similar they are in these different morphological characteristics, size, etc., color. And we organize them then in a 2D diagram that then when we look actually how they're organized is that we have darker species or darker images here, more transparent in the area. These will be large and elongated, so long and very big. And this actually will be round and small. So now what we can do is we can identify which are these ASV modules, which are these ASVs, which are the taxa that is there. Or for example, when do they peak during the year? So this is the day and the night samples, and this is the relative abundances of this module here through the year, okay? So, okay, we know now suddenly this group actually peaks during July and August. So they are the ones related to the decrease in the carbon export. We can get a table and we can say, all right, so what we have here? We have here these 11 calanoids, 11 copepod calanoids, 3 cyclopoida, couple ostracods, follow that name, ostracod. I will come back to it in a couple of slides. So now we know them, we can follow them in these ASV modules. But actually what we can do is based in these images, we can try to look, okay, we have these peaks, we know when they peak, we can try to follow these groups here within the images. So we know that when this module peaks or whatever, what we have then is they are distributed in the more transparent area or in the more round, okay? So then we can start feeding these ASVs or this molecular information into the PCA and trying to understand, okay, what happens when this module peaks? How does the community look like? All right. So we're trying to still understand and improve how we do these things. And why are we looking at this? This is actually an ostracod I was talking to you. I would like you to look what it's doing. This little ostracod was in one of our experiments, ruining the experiment, but doing something interesting. It has a piece of aggregate. So one of this snow is the marine snow that is falling down. And it's actually, ay, ¿por qué no tienes? Perfect. It's actually eating it. So then see how it's going to start playing with it, and it's going to start eating just the very, very brown. It's going to try to avoid the black particle. It's just specifically in the very, very bright area that is actually carotenes degraded. So it's still, there is a lot of food there to use. But then we'll see now diversity is influencing these ecosystems and the processes, because there are different species. One of them they are creating export, the others are just creating it and attenuating that. So then is the low export caused by low production, or just because there is a lot of these guys feeding everything that is passing by? Because we see that some of these ASBs, they have some specific ostracods peaking. Or maybe it's both. We have both big animals creating the big draft during when there is a lot of production, the big drag of carbon. And then when summer, then these guys peak. Then who are these guys? These are very transparent. In the soil scan, it's very difficult to see them, gatekeepers of the flax. So then we're trying to understand how this happens. Now this guy has finished the beauty, the delicious part. Now he's eating one of the black things that it doesn't like, and it's still eating. You can see how the food has gone through the gut into the stomach. And when it's done with it, it's going to kick it out and go. So actually, we're trying to understand not only the guys that are taking the carbon down, we're trying to understand which are the taxa that are actually preventing thus marine snow to reaching this capture zone where the carbon is removed from the atmosphere. Because once they eat one of these ones, they re-respire, and then the carbon is back in the water. And that is the part of carbon or things like that. And I want to show this last part, that is something that I'm starting to do, that is building the reference for the next generation. And the next generation, both of people and the data that we're getting, and what do we need to do to make these reference databases that we use or the museums accessible to everybody in the world. Okay, and leading this, but the person doing the work is Yuki Nimi, a PhD student I'm co-advising at Arizona State University, and this is funded also for one of the grants I have from the National Science Foundation in the US. So everybody is familiar, I suppose, with the term of barcoding. It's a program that was launched 20 years ago already. The barcoding of life that included a subproject that was the sensor of marine life. And for the zooplankton, there was a field project. Actually, that was the legal term, the sensor of marine zooplankton. This is a screen capture, and said this website is no longer being updated as of 2011. I was working there with Anne Buckling, working in these projects. Actually, this is a lot of my memories when I was a postdoc. But I suppose that what most people are familiar with of this barcoding of life is actually BOLD. It's indeed the BOLD systems. This is online database that people submit. You can submit a picture and an animal or whatever to this center in Canada, and they will publish it automatically in BOLD. And you can check your molecular sequences of your animals if they're in the reference, and you compare it to the image. And this is great. However, it's great to have the sequence and the image, but the images usually they are good, but they lack resolution enough to have a taxonomic use, or they don't have the right angle to capture the right characteristics. So they are not very useful when you're trying, okay, is this really the same species? So what happens when your molecular ID is different to the morphological or the other way around? So I'm thinking, how can we provide a database that can bring you a full detail image or model of the barcoding individual? So you have both. Because the databases are great when you have a full agreement. You take your animal, you think that this is the species, you put it in the database and spits you down, spits you back. Yes, this is the species, and you're like, cool, I was right. But what happens when there is a disagreement? You think you have a species A and brings you species B, or the other way around. So what happens? You don't know what is going on, because it can be misidentifications of the people doing the database or by yourself, or mistake in the lab, they cross labels. So you don't know what is going on. Can be really true cryptic species, or phylogeographic differences, natural species variation, maybe historical names that they are wrong, etc. So now, to be sure, we really need both the sequence and the animal, the figure of the animal, to be sure, isn't it? So now we are combining micro-CT scan, that is the same of one of these TACs, what happens in the hospital for you when you have a wrong knee or something very wrong in your lungs, that you put it in this machine that is like huge, they put you there flat. And they take a perfect three-dimensional model of your body, but we do it with tiny objects. This is the instrument. Both for we combine it with barcoding and museum archival. We have a resolution at the micrometer level. Okay, we are talking about 1 to 5 micrometers, depending how much resolution you want to do, because scans come from few millimeters with very high resolution to several centimeters with low resolution, but we can do hybrid models in which we do a high pass with lower resolution general body, and then we can do special areas at higher resolution. So we can do these hybrid models. There is a lot of computational magic here that Yuki is doing. I'm too old now to learn, so he's doing it. And the example I'm going to show you is the euphocytes of the Sargasso Sea. There are 8 genera and 41 species of euphocytes. There are several of them that come from a species complex that are difficult to ID by morphology, and they have, however, very different life, behavior, role in the oceans. We are looking also at the carbon flux. Some of them, they migrate, they get carbon flux strong, they take it deep to the ocean. Others, they stay in surface or subsurface, and actually they're going to be feeding all these particles that pass by. So actually they're going to attenuate that carbon flux, they're going to decrease the amount of carbon that's exported. But how can we take them apart if they are all of them so similar to each other? So we do this, we are trying to do the barcoding, we're trying to do the images, but we want to know which species we have there. Are they the same everywhere we are? Because also we're going to take that study out to other regions of the world. So then we do this 3D scan, and this is one of the examples of the images we see. We put the animal in the sample. We do a a lot of single slices of the animal. You can see one of the slices. And then we take that into the computer and we assemble a full animal as it is here. So I'm going to then zoom you in this image so you can see what we can see. So this is, for example, the eye of that animal. All right, you can see here the eyes, the multifacets, but this is, for example, anybody that is doing euphocyte taxonomy, I'm sure almost nobody is going to be doing here. We look at the lappet. The lappet is like a small scale that is in the antenna, that is this structure. And you can see the lappet perfectly, even you can see the hairs growing in the lappet or this spine. Even if we go to the carapace, we can start to see some specific small dots that we cannot see in microscopy. So maybe we can use both the morphological characters that we know that they are taxonomic to analyze them, or maybe you can try to find new ones. Because the advantage of these models is that you are not taking pictures of, all right, you look at the key, I need the 6th segment, the 5th leg, or whatever. I'm going to take detailed pictures of those. No, here, you're going to take details of all those and of all the parts of the animal. So we can look for new characters that might be actually the ones useful to identify between new cryptic species. All right. And it's useful for both external and internal organs. Here in green, in this green box, I have highlighted the photophore. So we can see where the photophores are between the pleopods of the euphocytes, and we can see the internal organs that they have. Forget about, for example, here we have the muscles, the gut, we can see many details. But this, for us, is the new method that we are trying to follow. Because then when you are even in Philippines, you can request the sequence and you can request this 3D image. And you can be looking at your own animal in the microscope, comparing to a 3-dimensional model that has all the details in your computer. You can see if you are really looking at the same animal with the same morphological characteristics. Okay. Other magic we can do is that once we have the model, we can separate. Okay, show me this second pair of legs or whatever of Stylocaron. So then we can extract them, and you will have the image of only that one, so you can see them clearly, right? Or if you have a key, an interactive key, you can say, okay, show me the second leg or whatever. So even if you are not sure what you have to be looking at, you click, and then on your model, that single piece will be highlighted, and then you will be able then to look in your animal what you have to look at, right? And I'm going over time. I'm sorry. So I'm very excited about this project. That's why I want to show you. So again, characteristics both established morphological characters and maybe new ones that you might need for new species. It's not limited to a set of pictures that somebody takes. You can see external and internal structures. It's non-destructive. So then you can take the animal, is back into the vial, into the museum, is there. We can get DNA from it. Allows from this museum archival, you don't destroy it. And this 3D model then is available for anybody. You don't need to zip the type. You can just take to your computer the model of the type. You no need of traveling, no need of shipping, and actually then it's accessible to more people, isn't it? And my other uses, phytographic variability, cryptic species, adding new references, so infinite, isn't it? I think this is going to improve and increase the use that we're giving to museum collections. Anyhow, a little bit over time, I'm sorry. Thank you very much for your attention. Thank you for the funders that we have. This is always important because, well, it's very difficult to do good science without funds. And thank you also to the Smithsonian Institution for the fellowship for Yuki, to my student, to go there and work with them. And again, the BATS program that has been going on for a long time in Bermuda. And thanks to the organizers and to everybody here listening to my talk. All right, I'm all done. I need to stop sharing.
Thank you very much. Sorry, there's my dog. Thank you very much for the very interesting talk. I guess a lot of students are very excited at the moment here at the audience. Just want to say that we have over 100 participants, both here in Zoom and also on Facebook Live. So I guess a lot of people are really interested in what you do. And I guess, yeah, so we have collected a lot of questions already, actually. So I hope you're ready.
Yeah, no problem.
Okay, just a moment. Okay, so one of the questions is related to the imaging. So what makes the individuals darker? You showed a PCA image
Yes.
of the dark versus transparent organisms, but yeah, the question is it because of the muscles or is it a marker? Or actually, my question is, could it be because the larger it is, the darker the color becomes in the imaging?
Both. So actually, we have some of the very big ones. They are very transparent because okay, we have a lot of copepods that they are migrators. So when they are strong migrators, they have to have strong muscles to swim up and down. Okay, so these are going to be thicker muscles. But actually, since they are arthropods, the muscles are anchored to the carapace. And to move those muscles without your carapace going completely collapsing, actually you have to have thicker carapace. And that is going to make your animals darker. So that's why Pleuroma massifias, that is one of the animals that I chose, they appear completely black in my images because they have very, very strong exoskeleton. However, when I look at some of the Rhynchalanus or other animals that other copepods that actually don't migrate, the carapace is extremely thin. You can see through them, and there's no problem. And they're very similar in size, even in thickness, or even, I don't know, salps or whatever. So it's both the size, but it's not only the size, because size was not actually linked to carbon. Actually, it's this darkness that we have that I suppose, or my theory, is that is linked actually to the ability to migrate. And then you have stronger muscles and a stronger carapace.
All right, thank you. So another question. One of the most dominant zooplankton are chetognats, mostly transparent but long. So where do they belong in the PCA image, dark versus transparent?
They go into the dark elongated. If you remember, they have a dark, and there was like long, large and elongated. So there are most of them in that region of dark and elongated. And they are not really linked to carbon export. Actually, I don't know if it's because they don't migrate that strong. We have always to think that copepods dominate the zooplankton, at least here. At least, well, here, not in Spain where I am, but in Bermuda. So they're about 60% of the community, 60, 70. So a lot of the responses that we see at the community level, is because of copepods, because of copepods, isn't it? So then the others are a little bit obscured. Also, we saw ostracods, we see salps, we see oncopleuras, isn't it? So it's clear when there is a group that has some impact appears in the analysis, and chetognats, they were not that abundant when we saw that.
Okay, another question. So I guess a lot of people got interested with your video of the ostracod feeding. So Yeah, so the question is, what drives darker images for plankton during spring bloom? Is it the food abundance?
Yeah, so when we have the spring bloom, that we have it actually in winter, February, etc., what we have is a lot of these diabetic migrators increase and be more in surface, let's say. So we have Pleromammas iphias, the other Pleromammas, many of the Auglaptilidae, etc. Sorry, Aritelidae. Auglaptilidae actually they are very transparent. Aritelidae, etc. And then we start to see them more abundant. They are coming up, feeding in surface and reproducing. We don't have any kind of blooming species linked to the spring, such as the Calanus or the Neocalanus or the groups that are in other regions of the ocean, because we are too poor. Actually, biomass, even when it blooms, is very low compared to their areas. I mean, we are oligotrophic. But what is happening here is all these dark copepods that usually inhabit the mesopelagic, and they come to surface at night to feed on during this diabetic migration coming up and down, is the ones that are increase the numbers, actually. So are the very dark copepods.
All right, thank you. So, we have a question from Neil Andrea. Hello, I enjoyed your presentation. I wanted to ask, how long does the computer generate a 3D model of the observed plankton?
Okay, that is okay, I suppose is the euphocytes. Actually, the most complicated part was the learning how to do it. And the complicated part actually is that it needs a very expensive video gaming graphics card. So it needs a top-end NVIDIA GPU. And that's something that even I'm struggling with because my computer cannot handle. So actually we're trying to use some of the common resources for research in the US to create a virtual machine. But if you have the right card, I suppose that in less than a day, once you have the images, in less than a day, you have your image. It takes a while to get all the images, and the higher the resolution, the longer it takes. Isn't it? It takes, if you want to get a resolution of about 50 micron, it's very fast because you don't need that many images. If you want to go to the 1 to 5 micron resolution, it can take a few hours to do the full scan of the individual, because we are talking about thousands of images even for a 1 centimeter animal, isn't it? So there is some machine time and then some computer time, but once you have it, if you have a good computer, it shouldn't take that long. And actually one of the things that we're considering is getting a video gaming computer laptop actually for the student, but I'm afraid that Yuki is going to use it for video gaming and not for this, but whatever.
Yeah, that's right. Thank you for that. There is a question here. Do you also record abundance data? Do you do it manually or using FlowCam?
So we get the abundance data with the source scan because the source scan is quantitative. The problem is that then we have these large taxonomic groups, so Calanel, copepod, etc. It's true that we can do, for example, if I have all my copepods already analyzed, I can extract very easily, for example, Pleromammas iphias. If you're interested in one species and you have a good training set, actually the machine, the artificial intelligence is very good at that. It's scary levels of very good at that, isn't it? You just need about like 5 different passes or 5 consecutive passes to extract all of them. So unfortunately, we don't do any microscopy. Again, I will not get funded to do it. I did it for my PhD. That was it. Really, I sit in the microscope and count. But unfortunately, nowadays, almost in the US, nobody's going to find you to do that. And this is my funding environment. I'm not saying that it's the right thing. I'm talking about what's my reality. I mean, it's not. I'm not here saying that anything is better than the other, but I get total counts with the source scan actually.
All right. Yeah. So, well, in the Philippines, we still count manually. So it's interesting, especially for undergraduate students. They have to learn how the plankton look like.
Exactly. This is something, and it's unfortunately is getting lost, the knowledge. And when we started the barcoding of life, several of my taxonomy friends and Ryuji, Dr. Machida, will know the same thing. They were against it. They said that they were going to kill taxonomy, etc. And we try to from the census, we say, no, we have to take care. This is just to help. And unfortunately, they were right in many cases. Many programs are like, well, we don't need taxonomy anymore because we have the molecular. And what happens? That we have species that will never be described. Recently, I was, I'm in charge of the Twitter of the sense of the World Association of Planktology, and they describe a species of copepod that is already extinct, for example, because the lagoons where they live, they are now completely degraded and there is nothing living there. So there are many things there to describe. I have a species to describe since 2006 in a bio, since 2006. So for the last almost 20 years. And I don't have time to do it. And it's very cool, like orange, and it has like a bump in the back, very beautiful. But I really don't have time. And the other thing is, it will not get me credit for promotion or for grants or for anything. So then I like to describe this species? I would love to. Is it good for my career and feeding my family? No. So then I might need to wait until I retire. Then my life doesn't depend on my papers, my publications to do it. And it's difficult. It's a difficult point to be. I understand.
Yes, thank you for pointing that out. Okay, so another question. We have a lot of questions actually. So what's the current status of barcode reference database for marine zooplankton taxa?
So we have a working group that is led by Anne Buckling, and we're trying to make it accessible. I can send you later a link, or I can, because I don't want to start looking on internet, but I can send you later the link because we're trying to take from GenBank, etc., and clean and give people actually curated reference data for zooplankton. And I think this is the most useful, and you can feed it into the most popular pipelines that are now for metamarket analysis. But the truth is that it's slowing. I mean, I can see that there is no much new information coming in. We have now a lot of sequences for Calanus, all these pieces of Calanus, or for Calso Calanus. I have many of those, etc. But since we don't have anybody working with, or not a lot of people working with very difficult groups, I don't see that progressing much further. So unfortunately, unless I see some groups coming actually from Japan or from Southeast Asia, even India, actually not so much from Europe or from the US, they're actually providing new barcodes and new species from those regions. That is great because they were the areas that were lacking more effort, just due to historical reasons. And that's amazing. But in other parts, I think it's starting to slow down, and it's going to keep improving very slowly, very slowly.
Okay, thank you. Another question, what is the advantage of using micro CT? Is it better than scanning electron microscope?
Well, the micro CT scan is because then you can take all the images and put them together and build a whole model, and the machine does it automatically, isn't it? So I don't get a single slice of the animal. Is that then I can take all the slices and stack them on each other, and then have the half animal. I mean, I'm talking about the image that I was showing there. That animal is about, I think it was one of the big ones, like 4 centimeters. So to give you the idea of the size of that. And then in the 4 centimeters, you can zoom in and go to the about 1 to 5 micron detail. So, and that is something, especially in those 3D models, I can rotate. I can see the full organ. I can take down the struct. Okay, give me this leg, take the leg out, or the, I don't know, the copulatory organ of the euphocytes is in the first pleopod. That is taxonomic for a lot of them. Is the only taxonomic character for many of the species. You can get the struct, and you will have the 3D model in your computer. You can rotate, and you can compare. That is something that the other microscopies does not allow, because you have a slice or a flat, but you have a flat image. You cannot rotate the animal. If a leg is behind the other one, you will not see it. In this one, you see everything that is there.
All right, thank you. We have a question from Sheldon of the Philippine Jellyfish Stings Project. He says, "Thank you for your presentation, Dr. Ria. I am just curious which specific taxa, perhaps genus or family of siphonophores were detected in your samples. How were they classified according to transparency, transparent or translucent?"
Well, first, I was not looking, and I haven't looked. I do community ecology, so I rarely go into look at the details of the species, okay, of the taxa, because that is not what I'm trying to do. I'm trying to understand global changes. So not a specific. I have paired with people with a specific interest in groups, and what I do is I extract the data and I give them to them, and they can do their own specific groups. So actually, I haven't looked at detail. I cannot say that. I can see, for example, that I can see porphyta, the blue button of the ocean. I can see where the larvae go because I have a signal of them. I have the adults in surface, and I have a signal of them in the very deep. That likely is the larvae. This theory that says that the porphyta, the blue button larvae, they develop in depth and then they come up. So I can see that in my data, but I don't know which species apart from that one, just because I was trying to help a friend. What transparency? Actually, they're very transparent. The problem with siphonophores often is that when they go into the net, they break. So then we have the bells. We have one set, what is the floating in one part and everything else in a separate one, isn't it? However, some of the siphonophore tentacles, etc., they go into the elongated and dark. Unfortunately, gelatinous, since this is samples from nets, gelatinous taxa that are big, they usually fragment a lot, and they're almost everywhere in the samples. So this is not the ideal sample for siphonophores, unfortunately. The images, the molecular is, but the images are not.
Okay, maybe we'll have one last question from an anonymous attendee. In terms of modeling and micro CT scan, what zooplankton group have already been modeled, except for aside from UFAUC?
I'm not very aware of that. I mean, I know there are people starting to do it because I was talking to the people at the Smithsonian Institution that is where Yuki is doing now the internship, and they're trying to CT scan everything. But I think this is even if it's not there, is the future. I know that for copepods that are very small, instead of using micro CT scan, they are using the laser microscopy, the confocal, because the laser is able to pass through the whole body. And they built a similar 3D model with higher resolution because the laser is able to do a higher resolution than a CT scan in less time, isn't it? And they create the same thing. They create a 3D model. My colleagues from Segenberg, they can even do a 3D, one of these images that they create in green, in red, and they put them slightly off, and then you put these color glasses, and you see them like three-dimensional. That is pretty spectacular. But the same thing, they can rotate it. So in copepods, small copepods, I'm sure I'm aware they're using the confocal laser microscopy. Jimmy Bernot, that is postdoc at the Smithsonian Institution, has been using also the micro CT scan to characterize parasitic copepods, because with the CT scan, you can characterize the external and the internal part without destroying the host. So I'm sure that they are doing that. But I'm pretty sure that this is going to be the future, or should be the future to most museum collections. The instrument is expensive, but I suppose that as everything once gets more used, the price will decrease and will become more available. That happens with sequencers. At the beginning, sequencers, there were one per university, and now they're left and right everywhere you can see one. So I suppose that will become more once it becomes more common and more accessible economic from the point of view of the price. I imagine that a lot of the different groups will become also CT scan. I hope so.
Okay, thank you very much. Very interesting talk, Leo. And I hope, I believe that you have inspired a lot of Filipinos today. Thank you very much. I think I should give you the floor back to the virtual floor back to Louis. Thank you very much again.
Thank you.
So yeah, we'll just make it quick. So I'll just sign of our appreciation to Leo's effort in presenting this topic. So we'd like to award the certificate to Dr. Leocadio Blanco-Bercial in recognition for his outstanding contribution as keynote speaker of the presentation entitled Plankton Diversity in the High-Throughput Era at the third lecture series on Philippine Planktology on August 29, 2023, signed by Dr. Sheila May Santander de Leon, the Director of the Institute of Marine Fisheries and Oceanology, UP Visayas, Christy Acabado, and then myself. Thank you, Leo, for your time, and we really appreciate your efforts. So hopefully we were able to like inspire new planktologists in the future, because yes, mentioned during your presentation, like less and less people are becoming interested like with the plankton groups,
Fortunately,
especially in taxonomy.
Yeah.
We have to. They're very important.
Thank you very much for the invite, Louis. I much appreciate this.
Yep. And then just last part, Christy might want to do some plugging when it comes to the different programs available in their institute. Christy, you're muted.
Yes, thank you for this. But actually the enrollment period is finished. But yes, I just want to introduce our degree programs offered in the Institute of Marine Fisheries and Oceanology. We have of course Bachelor of Science in Fisheries, Master of Science program in Fisheries Biology and in Ocean Science, and also we offer a PhD in Fisheries. Please watch out for announcements for the next academic year. Thank you very much. And also, please follow our Facebook pages, the Institute of Marine Fisheries and Oceanology, easy to find, and the Philippine Planktology, where we share a lot of information about recent updates on plankton research in the Philippines, and also the Planktonish Laboratory, where we share our exciting journey as we train our new students or the future of the field of planktology. Thank you so much. Thank you everyone for coming. I believe the evaluation link has been shared in the chat box. You may find it in the chat box, or if not, you can go to the FB Live posts on the different pages that was mentioned earlier, and then you can find the evaluation link there. Thank you very much, Leo, and everyone. To the Institute of Marine Fisheries staff and faculty for organizing this, for Dr. Louis and the students of the Planktonish Laboratory. Thank you very much.
Thank you.
Thank you, Leo.
Thank you.
I'll be forwarding to you the link where we usually upload this video, so you can have the copy and then share.
I will post it also in my Twitter. So
okay.
So then
Sounds
you'll get.
good.
Yes, please.
Thank you.
Please follow Dr. Leo on his Twitter account. Thank
Thanks,
you very
Leo.
much.
Have a good day.
See you.
Take care.
Sir Jun, we can end the live now. Thank you very much.
Take care.
Oh, maybe we can have a photo.
Okay.
With
No
the panelists,
Okay.
please. Yes. Elise, can you do the screenshot, please? Oops.
Yes, ma'am.
You
she's
are upside
inverted.
down.
Okay.
Okay.
1, 2, 3.
Okay. Thanks, Leo.
All right.
Thanks,
you.
Leo.
Thank you. Take care.
Bye-bye.
Bye.
See you around.